So I'm going to talk about the country-specific effects of neonicotinoid pesticides on honeybees and wild bees. So just to start with, we're going to talk a little bit about the importance of neonicotinoid for farming. Now, it's a very, very important pesticide. It accounts for 24% of the global pesticide market, an actually estimated value of possibly in excess now of 2 billion. The figure there is slightly out of date. It's mainly used as a seed dressing, and this, as you all know, offers a targeted delivery mechanism where basically the pesticide is taken up by the plant, and then the pests eating the plant get a dose of the pesticide. And for this reason, it's considered an important alternative to the older generation of pesticides such as pyrethroids. So it is a very important pesticide globally. However, it's very controversial, and lab studies a few years ago now have shown negative effects of neonicotinoids on honeybees and bumblebees. However, these have been criticised for being not really reflecting the real world. So on the other side of that, the field studies that have been done on neonicotinoid pesticides have often been small scale and relatively poorly replicated. And so these have been criticised for being inconclusive, with some showing no effect, some showing an effect. So it's really left regulators in the EU with a difficult decision, and what they've adopted is the precautionary principle, where we currently have a moratorium on the use of neonicotinoids on mass flowering crops, which you're all aware of. So in order to inform this debate and help regulators make a decision on the future of neonicotinoids, we really need statistically robust field trials reflecting the real-world exposure of pollinators to neonicotinoids. So that's exactly what we did. So the aim was to look at the impacts of field-scale use of neonicotinoid seed treatments on winter oilseed rape, on honeybees and wild bees. And the two products that we looked at are Bayer's Modesto, which is clothianidin, and Syngenta's Cruiser, which is thiamethoxam. So the trial that we undertook in 2014 and 15 was the largest study to date. It was the most ambitious study undertaken. It was in 3 European countries. So that's the UK, Germany and Hungary. Each of these countries grows a significant area of oilseed rape, and these countries granted us an experimental license to undertake the study. We applied the neonicotinoid seed dressings on commercial farms using the label recommended dose for each country. And all of our measures focused on key life cycle stages of honeybees and wild bees. So just to say a few words about governance and independence of our study. So the study was funded by Bayer and Syngenta to the tune of $3 million, and there was an additional piece of funding added by Centre for Ecology and Hydrology to further our understanding of the impacts of neonicotinoids on wild bees. CEH are an independent research organisation, and we reinforce this independence by the appointment of a scientific advisory panel chaired by Professor Bill Sutherland of Cambridge University. And this group had complete oversight of the study, ensured independence and excellence of the science. We followed the European Food Safety Authority guidance for testing the effects of pesticides on pollinators as far as practical for a study of this scale. We also had complete freedom to design and report the study as we saw fit, with all our protocols and data being openly available on our website or through other sources. And finally, as of the 30th of June this year, we published the results in a peer-reviewed journal, Science. So we've been very open, and we've ensured independence of this study. So experimental design, so statistically robust, statistically powerful, 3 countries, 33 farms, each farm growing between 50 and 60 hectares of oilseed rape, which is a sort of fairly typical area of the crop grown across the countries. The 2 neonicotinoid seed treatments were compared to an untreated control, so we effectively had 2 treated farms and an untreated control clustered in the same landscape and separated by 3 to 4 kilometres to minimise the chances of cross-contamination. The crops were grown under otherwise typical agronomic conditions, and all inputs were standardized at the level of the replica, if you like. So this took account of any differences in inputs. The moratorium on use that was in place at the time of the study further reduced the chances of contamination because no other oilseed rape in the landscape was treated with neonicotinoids. So that's the design. Key findings, and we'll go into some detail about the key findings in the remaining slides. So the study presents the first evidence of negative effects of neonicotinoid seed treatments on honeybees from a field experiment. So previous field studies have shown some negative effects on wild bees, but no one has shown a negative effect on honeybees from a field experiment. We generally found that clothianidin, Bayer's product, was more often expressed in the crop and resulted in more negative effects. And variation amongst the countries, that's the UK, Germany and Hungary, suggested that the effects of neonicotinoids were influenced by other factors, and we suggest bee disease and diet, and I'll go into that in some detail in a few moments. And the negative effects on wild bees, that's bumblebees and solitary bees, was linked to neonicotinoid residues from across the landscape, and I'll go into some detail about why that is towards the end. So they're the key findings, and we'll return to those at the end of the presentation. So just to give you some detail on the effects on honeybees. So how do we measure the effects on honeybees? We focused on two key time periods when we believe that honeybees are at most risk. The critical time is the strength of the honeybee hive following the winter. So after exposure, and they overwinter. So how large is that honeybee hive following the winter? And we believe this is important because this is the key time of hive bee mortality, and the fitness, the strength of the hive at the beginning of the year is important in its performance through that year and its survival longer term. And then the second key time is during oilseed rape flowering. This is when, if neonicotinoids are expressed, this is when the honeybees will be exposed to those pesticides. So two key times, overwinter survival and the oilseed rape flowering period, and that's what we'll talk about in a minute. So impacts on overwintering honeybees. So this is the first study, field study, to look at overwintering effects. So no one else has done this. And we found strong evidence of country-specific negative effects of seed treatments in 2 of the 3 countries. So this is what the data looks like. This is the size of the honeybee colony after the winter period. This is the number of bees following the winter. Firstly, you can see there's quite a large variation in the size of the honeybee hives between the countries, with the UK lowest, Hungary the highest, Germany intermediate. And then you can see that clothianidin resulted in significant reductions in size in Hungary, an average reduction of around 24%. So this is the clothianidin treated bees, and this is the untreated control, and you can see the reduction there. So a significant treatment by country effect there. And in the UK, we found a similar trend, but high hive mortality through the winter precluded formal statistical analysis. I should say that we took a very conservative approach to our analysis and only analysed data when it met all of the correct assumptions to allow that analysis. So we were very careful in the way we approached this. But what I will say about the UK is the median worker number in the UK was 0 for all of the clothianidin sites, you can see there. Whereas for the untreated control, it was above 0 for 2 of the controls, and for the thiamethoxam, it was above 0 for just 1 of the sites. So the trend is suggestive of a similar effect for Hungary, but we couldn't do formal analysis. For Germany, there's no difference, and we'll hypothesize some reasons for that in a minute. So the oilseed rape flowering period. So this is when the bees are directly exposed to neonicotinoids. So again, evidence of country-specific effects of the seed treatments. In Hungary and the UK, we found a negative effect of neonicotinoid use, and this was carried through to the following spring, that's the overwinter. In Germany, bees appeared to benefit from neonicotinoid use, but this effect was quite short-lived, and it disappeared after 3 to 6 weeks. It was not carried through to the winter, if you remember back to the previous slide. We'll give some possible reasons for that. So this is the data. So the negative effects of neonicotinoid, this is the untreated sites here in Hungary, and these are the treated sites. In the UK, there's a difference between clothianidin and the untreated, whereas these are the positive effects in Germany, but the data is quite variable, and those effects are lost after 3 to 6 weeks. They disappear. Oops, sorry. So, why are only some countries showing effects of neonicotinoids? So this isn't a smoking gun with piles of dead bees. You know, this is a more subtle effect of neonicotinoid seed treatments on pollinators. Why could this be? Now, we have some ideas, but I will say that this is not completely conclusive. Our study was not designed to investigate this, but we believe these are valid and possible, plausible biological reasons. So what are the honeybees feeding on? We looked at the pollen loads that the bees were carrying back to their nests in all of the country, and we analysed these under the microscope to determine the composition of the pollen load and therefore the composition of the bees' diet, if you like. And this is the percentage of oilseed rape in the diet of the bees in the different countries. And you can see that Hungarian and UK bees fed much more on oilseed rape. So between 40 and 50% of the pollen load was oilseed rape. So they are very reliant on this crop in the springtime, and so they may be more exposed to the pesticide. In Germany, where we found no effect over winter or a short-lived positive effect, it's only sort of 15% of the diet. So the German bees are feeding on different things in that landscape. They are therefore less exposed. So that could be one of the explanations for these country-specific effects. The other possible cause, we believe, is the level of bee disease. We used advanced molecular techniques and traditional microscopy techniques to look at the level of bee disease in the hives across the different countries. And you can see that in the UK, the hives had a far higher incidence of the varroa mites compared to Hungary and Germany, whereas the Hungarian bees had a higher level of the fungal parasite Nosema compared to the UK and Germany. So the point is the German hives were relatively healthy compared to either the Hungarian or the UK hives. And we believe, again, this could be another factor that is interacting with the neonicotinoids to give us this country-specific effect. So effects on wild bees. So how do we measure the effects on wild bees? We looked at two particular wild bee species, the buff-tailed bumblebee, Bombus terrestris, which is our most common bumblebee and is often seen in oilseed rape crops, and the red mason bee, Osmia bicornis, which is also seen in oilseed rape crops. Now, I should explain there are major differences in the biology of bumblebees and solitary bees compared to honeybees, and this will become clear in a moment. Honeybees are quite long-lived, can live for many years if well managed, whereas bumblebees are short-lived and they live for 3 or 4 months. Bombus terrestris, the colony, lives for 3 or 4 months, and new queens are produced in the middle of the summer, and the colony overwinters as a hibernating queen. So the queen then founds a nest in the following spring, and then the population continues from this point. So the production of queen bees is incredibly important to the long-term survival of bumblebees. This is the key measure, the key population metric, if you like. Solitary bees are even shorter lived, for a few weeks, and they overwinter as pupae. And so an egg is laid, and that hatches, and a pupae develops in a cell, and then that emerges in the spring, and the female bee then completes the life cycle by laying more eggs. So again, the production of eggs and pupae is absolutely critical to the long-term survival of solitary bees. So these are just shorter-lived pollinators, but the production of queens and pupae is critical to their survival, and that's what we measured. Oops. Okay, so neonicotinoid effects on wild bees. Well, there was no overall effect of neonicotinoids on queen or egg cell production. So this is the exposure with the treatment structure as it stood. That is different to the honeybees, where we did find that significant country by treatment effect. However, there was evidence of exposure to residual neonicotinoids from the wider landscape, and we'll talk about that in a moment. And this exposure from the wider landscape resulted in declining reproductive potential, queens and egg cells, as the concentration of neonicotinoids in the nest rose. So what we found was that the bees were picking up neonicotinoids that were not part of the study. Things like imidacloprid were turning up in the bee nests, and as far as we knew, this was part of we were in a moratorium, so it shouldn't have been used on mass flowering crops. It was being picked up from somewhere in the landscape outside the treatment structure. And as the concentration of the three neonicotinoids, imidacloprid, clothianidin and thiamethoxam, rose in the nests, the numbers of new queens produced by those nests declined. So this is a correlation. It's a significant negative relationship between concentration of neonicotinoids and the numbers of new queens produced. So it's suggestive that the bees are foraging over wider areas, possibly on different plant species, and picking up neonicotinoids from elsewhere in the landscape. We do not know the exact mechanism for this, but it is an interesting finding. Similarly, solitary bees, the production of egg cells is negatively correlated with this increase in concentration of neonicotinoid residues in their nests coming from the wider landscape. So, implications. The study has highlighted a number of areas of further investigation where we might be able to manage some of the risks around neonicotinoids. The first one, fairly obvious, is bee health. So the findings suggest these differences in the health of the honeybee hives between the countries. So one thing we could do to mitigate some of the damaging effects is manage bee disease better. And that is something that's very, sort of tangible that we could do. Diet, so providing a wider range of flowers in the landscape, especially in the springtime when oilseed rape is flowering, could reduce the exposure to neonicotinoids because the bees would have alternative flowers to feed on. So that would reduce that risk of exposure, dilute that exposure. So that's another possible thing we could do. Stewardship. The detection of residues in the wild beehives from across the landscape, and this is despite them being banned on mass flowering crops, suggests we need to look more closely at the persistence of neonicotinoids in the real world, in real arable soils. And that also suggests we need to look at how often we use them and where are we using them in crop rotation. Like all good things, they may have been overused, and we need to look at this and the persistence in the environment. Alternatives, well, we also need to consider what the impacts of the alternatives to neonicotinoids are on bees and other species. You're replacing neonicotinoids with another pesticide, probably pyrethroids, more frequently applied, and that is going to do some harm potentially to other organisms. So again, in order to inform this decision, somebody needs to look at the alternatives and the impacts. So I think I'll leave it there.